Ladies and gentlemen, welcome to MedSynapse podcast series, where we sit down with the brilliant minds behind the groundbreaking innovations and expertise. I'm your host, Dr. Nigar, and joining us today are visionaries in the field of healthcare, Mr. Deven Patel, the CEO and co-founder of Giostar, Dr. Anand Srivastava, the chairman and co-founder of California-based Global Institute of Stem Cell Therapy and Research, which is the Giostar, headquartered in San Diego, California, and Dr. Manish Kondapuram, a highly experienced radiation oncologist based in Hyderabad, where he specializes in advanced radiation technologies for cancer treatment, focusing on gynecology, oncology, breast, and brain malignancies. Welcome, doctors.
Thank you, Dr. Nigar. Wonderful meeting with you, and thank you for a very wonderful introduction.
Thank you so much for joining us. Now, a little bit more about our podcast today. Together, they lead a team that has developed a revolutionary technology producing mature red blood cells from stem cells in a controlled bioreactor. Tune in as we delve into the future of blood transfusions, discussing the incredible impact your startup is making on global healthcare. Now let's begin with our podcast. Could you please walk us through the key milestones and challenges in developing the process that allows the production of mature red blood cells from stem cells within a controlled bioreactor?
So thank you, Dr. Nigar, again for talking on this extraordinary topic of the blood production. And, the blood has always the scarcity in the market, and particularly after the COVID-19, there's a lot of scarcity of the blood. And so I started working in the field of stem cell science, I think almost 23 years back at University of California, San Diego Medical School. And I was always thinking how we can meet the challenge of blood-related disease. And if you know, the stem cell is the master cell of the body. The entire body is made of stem cell, but body is nothing but the combination of organs. And blood is also an organ. Okay, so and we have a lot of blood-related disease like blood cancer and genetic diseases related to blood, like thalassemia, sickle cell anemia, all these things. And there is no answer for these things. So I started thinking, is it possible to really differentiate the blood from the stem cells? Because inside the body, stem cell makes every single thing, as I told. So it makes blood also. But how to really make blood outside of the body using the same process which mimics the making of the blood inside the body? So that was the big question in front of me 23 years back. I'm talking, working at University of California, San Diego Medical School. Then first you have to take out the stem cell in the lab, then you have to propagate the stem cell, and then you have to think how you can really mimic that process which makes the blood in the body. So how to really replicate that in the lab? And that's what the blood idea came, and I started working on that. And to your knowledge, and for the people who is listening this, I feel so happy to inform that I could successfully made a red blood cell in the lab. And I did almost 15 years back, but it's a big process actually. And so this red blood cell was made exactly how the red cell get manufactured inside the body, basically, and or differentiate inside the body. And so I replicated the same process in the lab. Now we are having the red blood cells. So that's what we are trying to make it large amount in the bioreactor. So that's what we are trying to do it right now, to supply the red blood cell to the people so blood-related problem may be solved.
This whole idea of production of the mature red blood cells from stem cells within a controlled bioreactor sounds very interesting. Now, the elimination of human blood donors is a significant advancement. Now, how did your team approach ensuring the safety and universality of the produced red blood cells? And what impact do you foresee on addressing supply limitations and pathogen transmission risks?
So this is most important thing we have to understand. When we transfuse blood from one person to another person, there is always three problems we face. First, once you transfer from blood from one person to another person, you have to match the blood. That is the blood group, like A, B, O, AB, all these blood groups. Second, every time when you transfuse blood from one person to another person, there is chance of transferring the disease from one person to another person. Whatever scan you do for the diseases, that is not going to be sufficient at any time point. Where there may be some unknown disease you may be transferring. Third, there is a big scarcity of blood. And Deven will tell in much detail. So we collect a lot of blood, but it's still a scarcity. And after COVID-19, you see the blood transfusion or the availability of the blood is a big problem. So to solve this three problems, to matching the blood group and the transferring of the disease and the scarcity, we are trying to manufacture the blood in the control environment, the lab. So once the stem cell is differentiated into the red blood cell, that is going to happen in the control environment. So there is no problem of the developing any kind of disease inside the blood because it's in the controlled aseptic environment. And second, we have manufactured O negative, that is universal donor blood. And then third, because we are planning to manufacture a very large amount, so the problem of scarcity of the blood is going to be solved. So making this point, I'm trying to convey that yes, we have the blood, or we are going to manufacture the blood, which is going to be very aseptic environment. There is no problem of the pathogen because it's controlled environment, not inside the body, but in the lab. So the problem of matching and problem of transferring of disease is basically going to be solved.
Your technology aims to provide universally compatible O-negative blood type red blood cells. How does this expand the range of patients who can benefit? And what potential does this hold for simplifying transfusion therapy on a global scale?
So I will transfer mic to Deven. He can explain much better way. But in general, yes, we are going to manufacture the factory inside a bioreactor. So any amount of blood can be generated in the bioreactor, and then it can be supplied anywhere in the world because we have the blood which we are going to manufacture has very long shelf life. So it can be stored for longer period of time. So we don't need to worry. Once the blood is done, it can be stored for longer period of time, and it can be supplied anywhere in the world using the aseptic shipping technique, which we already have developed. So Deven, please tell a little bit more about our plan, how we can manufacture the blood and how we can supply the blood to different places.
So the answer to your question is how it is going to help in a different kind of medical condition. So there are a lot of diseases, metabolic disease or genetic diseases like thalassemia, sickle cell, they need a continuous blood transfusion. And there's always a problem of disease transfer, availability, all that is being solved because now we are creating O negative blood type, so no match required, and you have a pathogen-free, cleanest blood supply and unlimited supply, so they don't have to worry about availability and also the pathogens in the transfusion processes. That way, we solve the problem of the transfusion in the many diseases, also in the pandemic or also in the wartime. All these conditions, you need a blood and you need a blood supply right away. And as Dr. Anand said, the regular blood, you cannot store more than, I think, 42 days or so. now this blood can be stored for 2 years to 5 years. So there is no problem of storing it in a higher quantities, and it's a cleanest blood. And also it normal blood, when you and I donate, it changes several hands before it reaches to the operation theater. In our case, it's directly from our blood factory to the operation theater to the patient. So nobody in between. So we are disrupting in a very positive way and eliminating any chances of contamination, because it's coming from directly to bioreactors into the operation theater, and possibilities of storing it for 2 years to 5 years, and cleanest blood, no pathogens, and O negatives, or no match required.
Thank you so much, Dr. Srivastava and Mr. Patel, for giving us a brief overview on the potential this project can have in the future. It sounds very interesting. I'm sure all our audience would be interested to know more about this project. Now, moving on to our next question. With strong intellectual property and peer-reviewed scientific papers, can you highlight some key findings or insights from your research that contribute to the credibility, potential impact of your medical innovation?
So we have once, as I told, that I started differentiating the stem cell into the red blood cell. So during that process, how to really tell that yes, whatever I am doing is really correct or not. So I have to publish our findings in peer-reviewed journals, and so at least our peers can review my data, and then they can tell, yes, this data is having some significance, and then that has to be published in well-known journals. So I published when I differentiated the red blood cells in the lab, and I characterized those that yes, this is the red blood cell coming from the stem cells. And I published those data in very topmost journals in the field of stem cells called Stem Cells in 2003, 4, and 5, like that. So that is already published. Then University of California, San Diego went ahead and in fact they applied for a patent, and patent was granted by US Patent Office. So that is patented technology now, and those patent is transferred to Giostar, that is the Global Institute of Stem Cell Therapy. That is our institution. So that's what we have the right. We have published publications in the peer-reviewed journal that shows the credibility of our work, and also signifies that this particular cells which we created from the stem cell, that the blood cells, has ability to replace the red blood cells transfusion, and it can help with transfusion medicine basically.
Investing in groundbreaking medical technologies is a very crucial decision. Now, what kind of partnerships or collaborations do you envision to further advance and scale the adoption of your technology, making a lasting impact on healthcare worldwide?
The goal is to help a shortage of the blood transfusion all the time, all over the world. We collect about 100 million unit of blood around the globe, where you and I donate blood at Red Cross every year. About 100 million unit we collect. After collecting 100 million of unit of blood from human donations, we have another about 100 million unit shortage every year. And because of pandemic, people are not donating, so that shortage every year have gone from 100 million unit to 290 plus million unit of blood. So this technology will allow us to fill that gap of that 100 to 300 million unit of blood shortage every year, and allow us to partner with the blood banks, partner with the big hospitals, partner with the non-profit blood banks, and allow them to expedite the shortage side of the business. They are already collecting the regular blood from the people, but then they still are suffering from the shortage. So we're going to supply that with all those supply channels. And Dr. Manish, go ahead. You wanted to add to that?
Sure. I mean, so when it comes to voluntary blood donations across the world, most of the major blood donor contributions or the societies that actively come forward and donate blood belong to all these Western bloc of countries. It's the G7 nations. And when we talk about the rest of the world, to put a broader umbrella, the global south, so the Latin American countries, the South American countries, the African continent, so the 56 countries in the African continent, India, Southeast Asia. So the participation in the donor exercise is much more less. And it's a very stark contrast to what we see in the developed world. So because there's a lot of participation in active blood donations, they are able to fulfill their shortages much quicker. So to give you a number, it's 33.1 donations for every 1,000 individuals. So that's pretty high. And despite that promising number, there's a shortage. And a lot of times what happens is we say that blood is free. Now, when I mean by saying what blood is free, what I actually mean is that you don't have to pay for a bag of blood. But that being said, there's a huge processing cost. Now, you can't just donate blood anywhere and everywhere. You need a proper facility, you need a blood bank, you need a blood collection center. There have to be active personnel, and not just for the collection of blood, but for also the processing. The processing includes the key steps of blood grouping, cross matching, infection control, and it just doesn't stop there. So you also need to have storage facilities, you need to have refrigeration. Now, despite all of that, the human blood has a shelf life that is anywhere between 35 days to 42 days. So it's like roughly 5 to 6 weeks, after which you are supposed to discard it. So now we are not even taking into account the losses when we talk about the global shortages. Now, that being said, what we want to do is we want to, and from the process that I've mentioned so far, you can safely assume that from the point of donation till the point of receiving the blood, there are 8 people. So it has changed 8 sets of hands before it finally reaches to a point of infusion. And what we are trying to disrupt with our blood project is the blood is made in a bioreactor, it's packaged, and it's shipped right to a point of need. So there's a huge supply chain disruption that we're looking at. And because I said that there are multiple layers in our current existing voluntary blood donation system, even if there's an interruption in either 1 or 2 steps, the entire chain gets disrupted. So let's say if a blood bank has a refrigeration problem, there's a power cut, all your blood products have a viability and safety issue. And not to ignore the fact that despite our best intentions, despite our safest practices, there's always a risk of transmission of diseases. There's always a risk of a blood group mismatch as well. So now, given these risk factors that are inherently present in our voluntary donor system, it makes sense that we have a much more broad-based, homogenized, universal product, and we have it in abundance, so that all of these structural challenges, or let's say limitations that we have right now, are answered with a one-stop shop solution. And that's what we're trying to achieve. So it's not just a disruption. I mean, it's a market change. It's a marked change in how we practice medicine as well, because what happens is during emergencies, one of the game-changing thing is the window of opportunity, the golden period of time. In some cases, we can do point of incident transfusions. The only reason we don't do it is we don't have reserves of blood in the ambulances. So in certain cases, it could be an absolute game changer. So not just that, not just in case of traumas or accidents, even in case of people requiring regular blood donations like sickle cell anemia, thalassemia, a lot of compromises happen when we are unable to procure blood. So we delay transfusions despite the prescribed interval of giving it regularly. Sometimes a lot of these patients go without blood for a prolonged period of time. Now that's a huge challenge, and it affects their overall odds of survival as well. So when we have an abundance of blood, when we have a universal type of blood group like O negative, I don't need to worry about all these things, and you can focus your time, energy, effort, and attention on something much more vital, on the other problems that are critical in medicine. And again, in newborn babies, procuring blood is a huge issue. There's a lot of risk of transmission of infections from adult donors to the newborn babies. They still don't have a developed immune system yet, and because of this limitation, I mean, of course, we are still using human donor blood, but then there's always a risk of transmission of infections. So again, what happens is there's a poorer outcome when there's no adequate infection control in our blood products. So with this, because we are making it in a bioreactor, it's clean, untouched, uncontaminated. Even from the point of production to packaging, it's everything will be controlled artificially. So there's an artificial intelligence system that will be integrating. So no human contact, there's no interference, there's no risk of any scope of contamination. So it will come out as a pristine, clean, untouched bag of blood. And the only time that you'll actually break the seal of contact is when you're actually infusing the blood. So that's again much less risk than when we see the multiple events that happens from human blood donation. So all of these things are game changers. All of these things can massively improve the odds of survival of people, because they don't have to run pillar to post worrying about where they get their next supply of blood. And as you're aware, in OBGYN, a lot of surgical deliveries, they require a lot of reserves of blood. There's a lot of bleeding incidents. So again, having reserves of blood is very vital in improving outcomes. And that's what we're trying to do. It's not just, broadly speaking, a shortage we are trying to solve, but from that solution to the shortage, we have improved clinical outcomes. We may even see practice changing guidelines. So that's what we're looking at. And the impact doesn't just stop simply on getting the numbers of units of bags. It changes the entire landscape of how we practice medicine.
Thank you so much, Mr. Deven Patel, Dr. Manish Kondapuram, and Dr. Anand Srivastava for sharing your insights in the revolutionary technology that is shaping the future of blood transfusions in healthcare. This is an amazing way in overcoming the shortage of blood that we are facing currently. Thank you so much for your invaluable reviews and insights on this groundbreaking project.
Thank you so much.
Thank you, Dr. Nigar.
Thank you, Dr. Nigar.
Thank you so much, and wishing you a continued success as you continue with your innovative solutions, and looking forward to many more podcasts together.
Sure.
Thank you.
As we conclude, we want to express our heartfelt thanks to everyone involved, our guests, our audience, and the dedicated team making MedSynapse podcast a platform for meaningful conversations. Stay tuned for more episodes, driving innovation and progress in the ever-evolving field of healthcare. Thank you and goodbye.